So I'm here just to give you information about DNA testing. The DNA test, when people say DNA, there's not a form of DNA. So I'm here to mainly educate on what we're doing, what some of the DNA tests you've seen have been done, and just explain terms that everybody hears and really what they mean. And so to give you a little background, my name's Oostra. I'm actually a microbiologist. I've been doing water quality testing for 25 years. Started out with Other Health Systems. Went to Edge Analytical, set up their whole microbiology department, set up their Bellingham office, and then went on my own. I started Exact Scientifics. We moved to Ferndale a few years ago. About a year and a half ago, we started a molecular division. Janet Brooks and Casey are molecular biologists. They both have a master's degree. Jenna is actually a botanist in biochemistry. So she actually has plants that she has sequenced and patented. We work with practical informatics, and Ryan what he does is he's got his master's from Western. He's working on his PhD, but he takes sequence data and he's the one that sorts through it. So the main thing we do versus everybody else is we take a shotgun approach. So we sequence everything, and then Ryan will go through and sort through those sequences and tell you what's there. So I'll get more into that detail as we go along. Terms you should know. Before I forget, there's my visual. So we need to go over this a little bit. So when I was young, you bought jelly beans. So I'm going to be talking about jelly beans and jelly bellies. They all were different colors, but they all tasted the same. They were just sugar. That's really what fecal coliform bacteria is like. It's a test. It was a good test, and we're using it, but it really didn't give you any flavors or anything. It's just that indicator organism. Then we got the PCR. So now we got colors, and with these colors we got flavors. So PCR is a DNA type test. That was that next step that allowed us to get a little more, but it didn't finish the job. Jelly bellies, which are very more complex, it takes like 32 chemicals to make one flavor. So jelly beans are not easy, simple candy, but with jelly bellies, you got different colors, different tastes. There's a lot more to this jelly bean to this jelly bean. You don't take sequencing to really determine this. This is what sequencing gets into with DNA testing. The big thing that everybody has to understand is all this work that's being done with water quality, sampling is probably the most important thing, and I think you need to understand sampling. You go to a stream and you pull a sample and you turn around, put it down, turn around, take another sample, it's not the same water. So if you're thinking you're going to get the same results, it's not necessarily going to happen. So you have to understand that. So we always get the question is, well, yesterday they had a high count, today they didn't. Well, that water yesterday is down the Nooksack. So where you sampled isn't even close. So you have to understand sampling. You have to understand is when you take a sample, you take a 100 ml sample, and when you do a fecal coliform test, I don't look at that whole 100 mils. I'm looking at sections of it, which are dilutions. So when you see a count of 300, and I used a 10 ml sample, my count was 30. I didn't count 300 bacteria. So when in the world of microbiology, you deal with log 10 differences. So 10 and 100 in the world of microbiology, it's almost the same count, just because there is so much dilution in play in that. So you have to understand that. You have to understand is you take this as your water sample, and how many times I got to take this sample to get a black jelly bean. So the probability of grabbing that black jelly bean may take a few times. So just because you pulled one sample, the next sample could have it, the first sample could not. So understand sampling and how that plays into everything we do on the other end. So the terms you need to know. DNA is a carrier of genetic information in all living organisms. eDNA. So you'll see this term. eDNA is the DNA extracted from environmental samples. Then we get into water, we get into soil, we get into air. So that is what eDNA is. Gene, that's a distinct segment of DNA that is a carrier of information, so it determines your hair color, your eye color. So that's what a gene is. PCR primer, so that is a small fragment of DNA used to amplify a specific gene in a sample. 16S is one of the most used primers for doing bacteria surveys. So it's a big universal primer. You get into microbes, which are single-cell organisms, bacteria, another small micro-eukaryotes. Microbiome, so that's a big term that you'll hear. Microbiome is a community of bacteria that are in your gut. They're in humans, they're in cows. There are also microbiomes that are in watersheds. So when I take a watershed, it's a community that's in there. They're in soils. And what I'm going to do is, everybody looks around me. I'm going to switch around here.
Well, can I ask you a question?
Sure.
When I think of my intestines, that's to me a pretty clear microbiome.
Yes.
It doesn't go up to my stomach and
Right.
that sort of thing. When I think of an environmental sample, it's harder to think of a microbiome being that.
Yes, it's very, to really understand this, is when we talk to people what we're doing, they go, really? Because it is a complex system. It takes a lot of work. That's why you don't see, I'm going in really what DNA sequencing is now, but we're taking something that's applied in other areas and apply it to a more complex system.
Okay.
So.
Question, is 16S characteristic of microbes, or is it a gene that's in everybody?
No, it's more bacterial genes. Yeah. So the test we're doing, just to give you an idea, is this DNA sequencing we're doing now, it's not like we're making this up. This is a standard test. The federal government uses this test already. We actually have a contract with the National Park Service with Great Lakes using eDNA, where they take a water sample, and from that water sample, we're going to identify the amphibian species in that water sample. So they're using it to do profiles of fish, frogs, turtles. This is used all over.
Is that based on DNA they're shedding? Like
DNA
snails?
shedding, so everything's shedding DNA. It gets into the environment, settles down in the bottom of a stream, it's floating in the water. So they're taking those samples, and from that, instead of going out and trying to find all these fish, if you read up on newts in Europe, that's how they do surveys. They thought they had things that were extinct, and they're finding their DNA shedding everywhere in the environment, and nobody's seeing these species.
So like we lose
You shed.
skin scale.
Yes.
Skin cells.
Skin cells, hair, everything, fingernails. FDA uses this technique. Do you think of the Chipotle outbreak? So they took the bacteria strains from people who were sick, sequenced it, and then they started sequencing the products to see where the match was. First time ever, they had they went into a place in Portland, sequenced their environmental samples that was positive for listeria, and somebody got sick in Boston. First time in the history where they already knew the source of somebody that got sick from an outbreak of food and could link it right away, just like that. NOAA does this. So they're out taking eDNA water samples to identify corals. Hospitals, universities, this is a technique that's used. This isn't CSI. CSI doesn't exist. I don't put a little piece in a tube and out comes the results. There's a lot more work involved in this, so everybody asks the CSI. DNA analysis. So then we get into what is DNA analysis. So it's not one method. So when somebody says, hey, I did DNA, well, here's all the different types of DNA. So there is qPCR, DNA fingerprinting. Everybody knows about the Stavik incident that happened. That was done with the fingerprinting. That's how they found the guy that committed the murder back that long. So they had DNA from back there and fingerprint matched them up. Phylochip is what you hear is a microarray. So the EPA study that's happening with the ZAPS program is using phylochip. So there's all these different ways of looking at DNA. We're using next generation sequencing, where we amplicon, non-targeted, whole genomic sequencing. So PCR, what is PCR? PCR is the amplification of a gene or genes. They use primers, and we talked about what primers are, to target a gene or the genes of interest. And then it's either organism specific or universal. So if I'm looking for, I'll get into how universal versus targeted things. It's usually a presence-absence type test. qPCR. So qPCR uses PCR, but it also can quantitate during amplification, which monitors PCR in real time. So you see this graph that's on the slide there, and that's a real-time reading of they attach a fluorescence to the primer set, so when it starts amplifying, you get above a threshold which indicates a positive result for that specific gene. For example, these could be salmonella samples, and those samples went positive once they went over the threshold, and we had salmonella in these samples. Again, it's organ-specific, universal, presence-absent, semi-quantitative. The semi-quantitative is based on curve, so we build a curve with different concentrations of the bacteria in there, primer sequence them to see where they come out in that cycles. You see 0, 10, 20, so if I built a curve and I see something come off at 20 cycles, it could be 100. If it comes off at 40, it could be 1,000. So then we get into a microarray. So this is the phylochip. Phylochip is used in conjunction with the ZAPS program that EPA is running now on the NUCSAC and other different water studies they're going to be doing. What it does is takes PCR is used to amplify the 16S gene. So 16S is now universal, so instead of looking for E. coli, I'm looking for all bacteria. Then they take that and they do an indirect target approach using pre-selective sequences. They take this amplified material and they run it over this disc. It's a very small disc. That disc contains specific targets. So they run that water over the disc, the PCR amplified products attached to that disc, and then they throw a fluorescence dye on there, and whatever fluoresces tells you what I find. So it's a step up from PCR. What we do is next-gen sequencing. So what we do is we can do the amplicon sequence. So we get that PCR first, then we sequence the PCR. It could be one gene, it could be multiple genes. It's target or universal. It's one or many organisms. We can do a non-target approach. So this is what's unique about next-gen sequencing is I can just take a sample. So I could take the jelly bellies here, sequence everything, and tell you what's in here. I'm not looking for anything specific. I'm just saying I'm going to take it all. Let's see what's in there. Targeted versus non-targeted. So there's different DNA testing. Doesn't mean one's right, one's better. It depends on the question you're asked. So the question you ask is the method I'm going to use. So target approach. So here we get to our jelly bellies. So we get this jar, and we say, are there red jelly bellies in the sample? You can run a PCR or qPCR to answer that question. The PCR primer is targeting one thing, so we look for a specific target, and we're only looking for red jelly bellies. So the microarray technique can also do that. The sequencing the PCR product can also do that. But the answers could be different. So when we when we do the PCR, we can say, yes, we have a red jelly belly present in the sample. The microarray can say, you have a red jelly belly in there, and its flavor is strawberry. Sequencing, we say it's red jelly, strawberry flavored, and it's bean shaped. We have more information with the different things we used.
Can I go back one step?
Sure. Oh, we go back on this thing?
So if it's, what is it called when it's a red jelly bean, cherry flavored and bean shaped? You've got another step.
So let's put it this way. If we did red jelly belly, so sequencing, I can answer more than one flavor. So I can say, if you want to say it's cherry flavor, I use one flavor in this thing, but if I have red and there's 5 different cherry flavors, or 5 different flavors in that red, I can tell you all 5 flavors. I'm not specific to that. PCR can only tell you it's red. Microarray, if all they put on their little disc is strawberry, that's all they can tell you. If they added a cherry, they could tell you cherry. With sequencing, doesn't matter that question. I can answer it every time because I'm not looking for a specific thing. It's not a target. I'm looking for whatever's there. So, non-target approach. So you look into this jar again, and we can ask how many varieties of red jelly bellies are in the sample. 1, PCR. Use a universal primer set to target all red jelly bellies that are red. So we're looking for jelly bellies that contain red. So then we microarray the products from step 1, and we can sequence that products from that first step. So then the answer is, PCR tells us there's 8 jelly bellies that are a variety of red, including closely related colors such as red-yellow or red-orange. Microarray can then take it farther and tell us the flavors of those. Sequencing can tell us the flavors, and there also was a green-red watermelon flavored jelly belly because microarray did not contain that specific target. So this gets into that non-targeted approach where, since we're not looking for something specific, that's what this can do for it, the sequencing can do. So what colors and flavors are jelly bellies in this sample? We sequence everything. So microarray can't do this, PCR can't do this. You have to just take it and we sequence everything, and we say there's 27 different colors and varieties of jelly bellies in the sample. Each color corresponds to an identified flavor. Example, pink with red spots is strawberry daiquiri. Red with dark spots is strawberry jam. Blue with light blue spots is blueberry pie. So we can go on and on and on with those types of flavors because sequencing gives us all that information. So why PCR, why qPCR, why sequencing? PCR target, PCR requires knowledge of what you're looking for. So if we want to look for red jelly bellies, then we set it up to look for red jelly bellies. PCR, the thing with PCR you could require a secondary confirmation because it is really a presumptive look at it. PCR can have some false positives because if we're doing red jelly bellies and you tell me I want to look at red jelly bellies that are strawberry, well, if I only use red jelly belly, I may get the cinnamon ones also there. So I get a false positive reaction. The good thing about it is PCR takes less effort than sequencing and is cost effective. The results are presence absence, but it can also be semi-quantitative. Whole sample sequencing. Sequencing just gives us that more information. Red, very cherry, red cinnamon, yellow and white, buttered popcorn flavor. It takes more effort and it costs more. You need the database with references for colors and flavors so that you can identify what you find, and I'll get into that later. But there are many different questions that can be answered using a large amount of data produced. So when I sequence it, not only am I sequencing the color and flavor, but I'm getting all the other information. Was it also bean shaped? Was there a dent in it? Was there anything else that happened with it? Because we're gathering all that information. So what we're doing is we'll get to Scott Ditch, which is what study we're doing now. So this is, we call it phase 1. So we're using eDNA sequencing to evaluate the microbial community of Scott Ditch. We're looking at input source monitoring, where we're looking at the microbial community of the inputs, such as wildlife refuge, which has a flow stream going next to it, a berry farm runoff, you have housing development runoff, urban runoff, and are they the different or the same? So we're going to sequence them, and can we see differences? Is there a unique indicator for each input where we can actually track those sources down the stream? That is one of the steps that we're going to try to do. So we're also doing multiple methods with this. So not only are we doing the universal primer sequencing everything in those waters, but we're also taking fecal coliform samples at the same time and sequencing that information. And what we're looking at is, are we going to see anything distinguishable by just looking at that fecal coliform results? To give you an idea, as we sequenced the samples from Scott Ditch, 16 samples, took 24 hours to sequence them. The computer's running for 48 hours to sort through all the data just for those samples, and it was still not done today. So that's a lot of data that we're generating from this test.
Question?
Yeah.
Regardless of the library, are you expecting to see differences that you can discern statistically between sites? Or will you be able to, without a library, see that you have different communities?
Yeah, I'll show you some. So we actually did this for WSDA, and I have the actual screen that will pop up and you'll really see it. So we get into fecal coliform analysis. So fecal coliform is just an indicator test. It can't tell you where it came from, who's at fault, anything like that. Methods used, multiple tube fermentation, membrane filtration. Most people see membrane filtration, it's a CFU per 100 mil result. That's used to indicate contamination. So the blue color indicates what we do now. This is the test. It's fecal coliform. So the next step is, let's sequence these fecal coliforms. Now fecal coliforms are really just a group of 3 main bacteria. Within those bacteria are different species, genus. So with whole sample sequencing of the fecal coliforms, we now are able to distinguish what they are. So was it a Citrobacter? Was it Klebsiella? What type of Klebsiella? Was it E. coli? What strain of E. coli was it? Was it 0157? Was it 0126? So we can get all that information because we're doing a whole genomic sequencing. We're doing shotguns, so we're not amplifying anything. We're not throwing any bias into anything. So that will be able to break that down. So we're hoping that we might see differences here, which will really change how we take fecal coliforms and move on from that.
That's a quick question.
Yeah.
In terms of that. So when you think about doing the fecal coliform versus sequencing, do you cross-check with, say, how many colony-forming units you have, and how many sequences you get? Meaning, you always sequence everything that's in there.
We're going to sequence everything in there.
And you expect a complete coverage in terms of you will know what all of those things are, even have unknown sections, or
It matters how much what the question is. So if you want to ask the question is, what are these colonies? Then I sequence the individual colonies. If you want to say is, what is this population? I'm sequencing the whole plate. Because the trouble with membrane filtration is you look at it and you say, okay, somebody said that one's blue. Someone says this is blue-white. I mean, one person may count the blue-white as a fecal, was it or not? Did this fecal coliform sample, did this E. coli, for example, change the media blue? Or was it inhibited somewhat, so it never changed to blue, so it didn't get counted? So as much as you want to use membrane filtration, there are a lot of variability in there. Stress organisms thrown through a membrane filtration can die off. So to pick individual colonies, we could do it. I guess is, if that's the question you're asking, is which ones are what?
no, the question is more, so you do a fecal coliform test and you get a number of colonies.
Right.
Right. That's fine.
Right.
And it's a good indicator for how much stuff is in there. So when you do the sequencing, can you expect that what you will be sequencing is all of the stuff that's detected in the fecal coliform test? So what I'm trying to say is, or are you missing some things when you sequence?
I won't miss things, but I will, since I'm sequencing everything, if there's colonies on there that are not fecal coliforms that didn't turn blue, those will show up in my sequencing event. But I won't miss anything. Because if it's there, the sequences will come out.
If I could back up a bit, when you're looking at things under the microscope and you culture them, what color they are, how numerous they are, that's how we figure out how many bacteria there are in the water sample.
Yeah.
It's an imprecise science.
Yes. So here's the EPA study, and I brought this slide to kind of show you what they looked at. So the far right column, those colors indicated the number of we'll call them species, indicators they were looking at. So, you got your avian, your human, you use Bacteroides as the main thing, ran PCR. So you can see the 1 through 8 were the different locations, and each color indicates something they found. Red may have been avian, yellow could have been human. A lot of Bacteroides, no species, a lot of negatives. But that kind of gives you an idea, of, just by you looking at that color, of what they did. Now to compare, that's the WSDA sample that we did with whole genomic sequencing. Same color scheme is all those colors indicate organisms we identified. So since we took this and we did a universal 16S amplification, and we sequenced everything that showed up there. So we looked for anything that would sequence and come through is in that chart. So as you can see, that's a lot of information. That's a little different than what you saw before. So
Would you back up and show us the EPA again?
Middle one's
Ah,
EPA.
there we are. Okay.
So to show you, this is fecal coliform after sequencing. Before sequencing, it's just blue. EPA is they use a specific targeted set. So that's what they got with their target. So that's what they looked at. Compare it to what we're doing.
So each one of your vertical lines is a different sample location,
It's a different sample. That is, yes, each one is a sample.
and you haven't tried to match samples from similar areas at this time.
Well, this is, WSDA is pouring through all this data. So if you can see is, there's actually some of these samples had a treatment, post and after treatment. And it's very interesting to see the difference between the two. Is one population is really a lot, and then they did a treatment, and you see a less population in there. So some of those, the first two are actually controls, but there are some that are in the middle there where you see a big green, brown, small blue was post-treatment. So the sample before that had a lot more color in it. I mean, you see some of those with all those individual lines. It's a lot of bacteria.
What do you mean by treatment?
They did a water treatment. They may have done a chlorination. They may have done a filtration.
How?
These,
Go ahead.
are these samples different treatments of samples from Scott's Creek, or are they samples of the plates that
So
you did?
that far one,
Yeah.
WSDA, has nothing to do with Scott Ditch. They went out and took samples throughout the county.
Well, no, I'm talking about the far one.
Yeah, so yeah, the far right one
Yeah.
is a WSDA went out and sampled different surface waters and wells and brought those in, and we did a bunch of tests. Sequencing was one of those. That is the data from those samples. Those were creeks, wells, and different things. Yes.
Wow. How do we pull this back to identifying at the source? And I see a similarity between the EPA study and what you're doing with libraries.
Yes, so we're going to get to that. So, but just to give you the data generated from what we're doing is completely different than the others. So what's next? Okay, big thing is I need a reference databases to compare the waters to. So all that data I got out of those water samples are things we can compare themselves to, but I can't go take that data and compare it and say this is the source, because nobody's done it the way it needs to be done. So what we need is to go out and identify sources that influence the watershed. We need to do a year-long sampling of wastewater treatment plants, dairy lagoons, septic samples, riverbed sediments. Are these natural populations we find upstream? Are they natural populations as once the river go down that are in the sediment? All this needs to be done. I've just listed a few. There's a lot more. But without the reference database, I can't tell you. Like a jelly bean, you cannot identify a jelly bean flavor based on its color unless you had something to reference to. So they always give you this reference table to indicate the taste. So if you pull the yellow and white one out, it doesn't mean it's popcorn flavor. It could be caramel corn. So this has to be done to make genomic sequencing thing. And what we're proposing to do is take that, not only take those references and start doing whole genomic sequencing, but also do fecal coliform sampling on those and sequence this fecal coliform and say, is fecal coliform a good test? Can we tell anything from it? And maybe we find out is, yes, there is specific strains that are unique to each location and we can use it. Or we find out is, there isn't things that are unique, so it doesn't really tell us anything. So that should be done. So we don't know what we don't know. So the big thing here is it's a mass amount of data. This data lasts forever. It doesn't go away. Questions we haven't thought of to ask may be answered in the future. Because again, as we're generating things that nobody's looked at, just because it hasn't been applied.
So as you build the library, you can look back. and see this is
Right,
where the
so-
sample came from.
So I have all this data from WSDA sitting there. A year from now, I could go in and start looking at, if we built the reference database, take it and start comparing it and looking. So the great thing is, it's comprehensive data, allows for reevaluation in the future. So since we're not looking for one thing, we're looking for anything, We may see something, we have no clue what it is, and 10 years from now they go, oh, we know what that is now. Let's go back and look at that data and see if it was there 10 years ago. The other thing is not relying on historical indicators. New indicators can be found that could be tracking. So one of the things that could happen out of this is we start doing this database and we find out is these are unique species to each source. So we're going to build a method for these species. So because we know they're unique, so let's look for the uniqueness, not indicators that are not unique. Questions?
Alan,
If you can back up 3 to the different sample locations, or that one, next one, sorry, that one. Are you assuming that there will be more similarity between the output of septic systems than difference, let's say, similarities between septic systems, so you'll be able to distinguish septic systems wherever you're sampling water? In other words, it's different than, the signature is different than Berry Lagoon, Peak Bog, Wildlife.
Yeah, we don't, like I said, it's, nobody's done that, so we have to build this database. Worst case scenario, we start doing this, and we come back and say, there is no difference. And then you get into is, okay, you can't monitor it. So what are we going to do besides the standard fecal coliform test?
Yeah, this is all very interesting to understand what's going on in the watershed, but we do have to keep a focus on the fact that for shellfish bed opening criteria, you're looking at fecal coliform, and I guess for DOE has a standard for E. coli, and I'm not sure whether or not they have one for fecal coliforms in the freshwater system. So there's a difference.
Right, but we're not talking about regulation, I understand. I actually, when I was with Outer Health System, we were the only lab in the state, actually on the West Coast, that was going to get certified to do shellfish beds. I had somebody out from Boston and went through it all, through everything, but the state offers it for free and I can't compete with that.
Well, they don't do it for free, I'm sorry.
They don't probably anymore, but they used to do it for free until there was no market. But we're not looking at this as a regulatory issue right now. It's way too early. This is more of a monitoring. Can we help the shellfish beds? Because we can now say, is there a tracking source?
We can use this information if it all works out. And I've looked at some of the mathematics, and that's way beyond me to compare this stuff. But you can then inform what is in fact in the shellfish bed samples as being different from what they think it's indicating.
Well,
you can start on that process. And,
right.
So you're getting the infectious agents or the contaminant agents separate from the others. You can separate those.
Right. So what we could do, and I haven't even talked about, is not only can, I'm not talking to apply it here. Let's apply it to else things. I can do what they can't do now with this. I can go out in the middle of the ocean, take samples, start looking at indicators, and work my way back to the beach. Are those indicators showing up on the beach? Are they showing up in the shellfish? Because now I'm going to go to the shellfish and sequence the shellfish. When you talk about Lummi, I'm actually working with a shellfish company on Lummi Island that had a mortality rate last year. So we're looking at sequencing their issues and see, was it virus, was it bacterial, was it chemical? Lummi's had an issue with their fish hatchery. This could be applied. We could take those fish and start sequencing, looking for viruses. We can look for bacterial infections. Was it something else that caused it when that was a problem? And since we're not targeting anything, we're doing whole genomic sequencing, we're not saying, what do you need to look for? Because that's what the question everybody asks. What do you need to look for? We're just saying, we'll take it, we'll sequence it, and we'll tell you what we find. So that's one of the things that came with applied. It's been applied, the Fish and Wildlife did this. Ryan used this method and went through their system, found a contamination issue that was killing off the fish, and we found the source by sequencing different spots in their hatchery water.
I don't want to dominate questions, but I've got two. Other people have them.
Yeah, I have a question.
Yeah.
Are you expecting that just to simplify it, the color profile that you're seeing, if you see that in unknown sample, it's an indicator that if it looks like the original source, is that telling you that it's an indication that that may be the source of that?
Right, so what we're looking at could be one thing. So you could do the sequencing, we find a unique indicator that is specific to each source, and we go that way. What we find out is each source has a unique microbiome, and we know that these 10 things come from a septic system. These 10 things come from a waste treatment plant. These 10 things come from a dairy lagoon. So when we see those profiles out in the stream, then we say is, you know what? This profile is showing up here. Your source is here. So then we can go back and say, okay, you guys, there's something here because we're seeing your marker out in this watershed.
But that would be less likely to be informative if it were a mixture of contaminants.
Well, then if it's a mixture, we can look at profiles, and with the amount of sequence we do, it's possible that we can eventually look at ratios and say the ratio of this profile to this profile is indicating that this one is the main culprit. So it is possible. Somebody asked today is, can you quantify? It's not a yes-no answer right now, because we don't know. We know what we possibly could do, so if we found a unique identifier, we set up qPCR and now we're enumerating.
But a unique identifier is a key. If you don't have that, and you're just relying on profiles, it's a different story.
Well, profiles, but then we can look at ratio of the number of sequences for this profile versus the number of sequences for this profile. And there is ways to do it if you start wanting to know that question. It's just going to take more work. Right now we're just going, is it there or isn't? But there is a possibility of doing a ratio or percentage look at it, but it's just going to take more work to develop. Once we develop the database, we may say is, hey, we see that this is the profile. So now we're going to set up the profiles and we're going to start building this curve, and we're going to run lagoon samples, and we're going to run them at different dilutions and look at our ratios at each dilution, so then we can do a semi-quantitative approach.
Okay. Andy, can I ask one more?
Be my guest.
So once you get enough data to start picking this apart, is it going to go through some sort of a peer review process before it becomes something that, we're going to apply this to a study and we're going to start talking about it publicly?
That's our goal, is to do peer review.
Right.
If we're going to do this, we want to, there is actually money that was designated to do this database.
Mm-hmm.
It's sitting at the Conservation Commission. So, don't know if we'll see it or not.
Okay, thanks.
So, yeah.
Alan?
Yeah, it gets back to my original understanding of the thought that was there. You get a sequence and a series of sequences that would maybe characterize the proportions of different elements on a farm field, of different types of farm fields.
Yeah.
And then you would look in the water sample and see if you could take out that unique combination of samples that would suggest that, okay, a certain proportion of this is probably related to the farm field, a certain portion related to the forest area. Is that where you're going, I guess?
The long term is if that's a possibility. If we can say is if we build these profiles, let's say it ends up being a profile, and there's a microbiome profile, and we look into a watershed, and we're seeing different profiles, and we notice that when we sequence the sample, the number showing up for this profile is 100, but this profile is showing up at 10,000 counts. So our sequence ratios could indicate is, well, this one's way higher, so it's telling us this is more prevalent in that sample than this sample. Again, is to get there is without the database and knowing, it's a hard question to answer until we've done this research. But that would be the next research.
And I guess just the last thing, even though you say it's not related to enforcement, if it wasn't for the regulatory enforcement, nobody'd have a damn bit of interest in it.
Right. Well, yeah, yes.
So vacate while the sun shines.
Well, right, but what I'm saying is right now there is an account enforcement, yes.
And so one of the challenges with the library-dependent DNA methods earlier on was that if you didn't have a local library, you could have variations in that DNA, and so the correct sources weren't being identified. How do you see the progression of this? Because it seems like you're kind of needing to create a library that you'd be comparing samples against as well. Do you think that you're going to have to continually update that library for variations, or?
Right, so we look at building a library, it has to be at least a year long. For example, when we laid it out, we would take 10 lagoon samples a month, composite them to one sample, so we account for the variation. Because again, if you have one farm that's a Jersey farm feeding corn silage, another farm that's a Holstein feeding haylage, their profiles are going to look different. So we got to be that database to understand this reference database. The flow cell to run the sample is about $200. The reference database flow cell to run the sample is $1,100. So because we need that depth, we need that sequence information. If we put any holes in it, it's worthless. So it's not a cheap expense to run that database. That's where we're going to put all our effort and money, because you need to have a robust database to compare to. Right now, that conservation district has some money for east side, some for the west side. We would say is, okay, if we're doing, say we got both studies, you could take that data, parse it separately. You could take that data, put it together, and start building. We would say that you would always be adding to this database. If we did this in Whatcom County and Skagit says, hey, we want to do the same thing, I'd say, send me some lagoon samples. Let's see if they're different. And if they are different, then we add it to the database and we build the database a bit more. Again, is with that data, is we can parse it out. Maybe there's a Skagit profile, maybe there's a huge profile.
Cool. Right?
Will you just clarify what you're doing with the Scott ditch? Because I think that there's some value in, you don't have a library yet.
We don't have one yet.
But you can learn some things from it.
We can, because what we're doing is we're looking at what we call inputs. So there's Scott ditch, and then there's things flowing in. We're calling inputs. There's an urban runoff. That's an input. There's a wildlife refugee that's out here that has a water coming from it, berry farm. So we're looking at those inputs, sequencing those and saying, are they unique? Can we tell a difference between them, or do they all look the same? So that's where we're starting. What do those inputs really look like when you sequence all the microbial profiles from there? And again, is without knowing anything, we can say is, we found different indicators that are unique to each profile. Do we see those indicators in the ditch? So then we'll go look to see if we see any of that. So it's what we call a phase one, because what that data is, we can build, once we build the reference databases, we can go back. But it is going to indicate is, can we really differentiate the inputs and sources with these microbial profiles?
Can I ask what might be a simple question?
The other thing we're doing with that is that fecal coliform. So one of the big things is, pie in the sky, greatest thing that would happen is we sequence the fecal coliforms and we find out there are unique strains. And we go to our database, and in 3 months, we look at it and say, we've got unique strains. We don't need, we still, I would say, still continue the database, but maybe we don't need to sequence the water. Maybe we just sequence all the membrane filtered plates, and they send them in, and we sequence them. It says, you got this strain, indicates this.
Like OSS or wildlife.
Right. And since we're doing non-targeted approach to sequencing, what happens with targeted is you use a set of primers, you have a bias. And that bias limits your report. So with a non-target approach where we just sequence it, we can get down to strain level. Once you target it, you lose strain level type information. So if I see an E. coli, it's E. coli with a target. If I see non-targeted, I can see E. coli 0157, 0126. I go into more detail, and that strain level is maybe where we find information that will help us.
If I can, I want to try and encapsulate this in my brain. The non-targeted you're doing is looking at provenance, and I think of a sommelier, or I never get the word right, people that can take a sip of wine and tell you what year, from where, the soils, and all that stuff.
Yeah.
They nail it. That's what you're hoping to do with this non-targeted, is you're going to have that level of specificity.
Yes, that's the non-target to get down to that, yes.
What you described with fecal coliform, and it's different than what you described for PCR and what I think of as a CSI type approach, or the
Yes.
O.J. Simpson, where you know the target you're looking for, so that's what you go to amplify in your sample.
Right.
Can you elucidate on that a bit in terms of our problems not being able to tie a specific animal to a specific contamination event in a creek? How difficult that is?
Yeah, so part of it is so if you're looking for a specific thing, how prevalent is in the water is one thing. So you can amplify all you want. If it's not there, it's not going to show up. So that's part of the issue. So for looking for one specific organism doing amplification, is the black jelly bean in the jar. How often am I going to grab it to find it? Because with some of the sequencing, we're not seeing E. coli very high. So some of the sequencing, our initial ones, we had no E. coli come out of the sac when we sequenced it, because it's low level and you really need to pull it out to look for it. Once we use primers and you start looking for it, yeah, you'll find it. But the prevalence of other organisms and other things that will influence your recovery is part of the issue with PCR, because you're looking for a specific thing. If it's not competing, it's not there.
One step back, if you were sampling for vertebrate cells, would that be a worthwhile exercise? This is all associated with microbiomes, associated with animals, perhaps. Could you identify a cow abundance, or a sheep abundance, or a raccoon abundance from your sample if you had the right primer?
Right, you could. So
Before you answer that, we've got 3 minutes left.
okay, so to give you a good idea is when we first started, we thought we could shotgun sequence the water samples themselves. We found out there's not enough DNA.
Okay.
So we don't get enough recovery. So we said, okay, what's the most ideal? Because ideally we thought if we sequence everything, not only can I tell you what bacteria is there, I can tell you, did they eat hay? Did they eat corn silage? Did they eat blood meal? I mean, we could go down to that much detail because we looked for everything. But what we found out is the DNA that you take out of a water sample, we did 3 liters, and there's still a low amount of DNA. So you had to amplify something to get it out. But you could take any of that and say, I do a COX-1 versus a 16X. I'm looking at a whole different set of genes. So you could use other universal type primers to amplify other things. So you could do plant tissues. You could do like they used to look for frogs. We take a frog DNA primer set, we amplify and look for frogs. That's what they're doing for the Great Lakes.
If I could jump in, I'm loving this conversation. Are you willing to stay late?
Yeah, I'm fine.
And other people want to stay late? Thank you, but we've got a minute and 10 seconds for our normal meeting, and if people want to bail, they can.
Yeah.
I know next to nothing about cows and how quickly, or what might cause their individual microbiomes to turn over from sickness or whatever. Is that a thing? Do herd microbiomes change over time, or?
I would say that it depends on your foods. I mean, if a farmer is feeding alfalfa, and that's all they're feeding, they will get a specific microbiome because of the feed and everything they use. And if they're feeding probiotics, if they're feeding mineral contents and things like that, that will affect what shows up. They switch to a corn silage. corn silage has a different bacterial load. So that bacteria will start showing up in a different cow. So one of the things we talked about when we were looking at whole genomic sequencing was that we may have an issue where we see a dairy problem in the watershed. You know, we were thinking with the whole genomic shotgun sequencing, we could take that and say, oh, we sequenced it, and now we found out this sample has a lot of corn silage in it. So the farm that's polluting is a corn silage farm. And we go look to say, is there a farm in that area that has corn silage? And maybe this wastewater treatment plant, maybe Ferndale's is completely different than Linden's, and we say, we got a human issue, let's go look at the different ones and see if one of those specific primer sets shows up there.
Yeah, a lot more vegans in Bellingham.
Right, so you might, I mean, that's the thing, is everybody is different, it's a different population. So that is possible.
Okay. The amplification, I'm surprised you weren't finding enough DNA in your water samples. Is your amplification selective, or can you do everything comes up and you're reading that?
Well, the 16S it's every bacteria has 16S. So we're using one that every bacteria has. The trouble is then it's only bacteria. We lose the plant information. But we still have some of that DNA left over because we're not amplifying all that extracted DNA.
Okay.
So part of our study for Scott Ditch is not only are we doing a 16S, we're doing another indicator to confirm what we find. So we're going to have leftover, and we're looking at another universal. There's a lot of different universal primer sets. 16S is the one that everybody did. It's what's mostly published. So it's one that all the information out. But there are other universal sectors. So we're going to run another sequencing event using a different type of primer set as a confirmation step, and to get more detail.
Ask another geeky question? No? I would imagine with the results you've gotten so far without a library, you could do cluster analysis or some sort of statistical analysis and start to see separate provenances.
Right.
Are you pursuing that? And you're looking to correlate those to what's upstream?
Yeah, I mean, that's the thing, is that the beauty of it is we have so much data. This is what we're looking at, is we'll have so much data that we can't think of everything to do. One of the things we did with the one WSDA sample is we did a 3D image where you start looking at things and you rotate it. So when you look at it at one angle, you see all these dots here and here. You turn it, oh, these all line up. You turn it this way, oh, these all line up. So all that, I mean, there's so many different ways to look at the data that's the beauty of this. It's so much data and there's so much you can do with it that we haven't even thought of all those things. So
That being critical.
right, and I know we have somebody from Western that really wanted to do a lot of the statistical evaluation. That's what Brian is. I mean, he's the bioinformatician. Like I said, he's getting his PhD in computer bioinformatics and looking at all different ways to look at data. He does this. He was going to be here tonight. He's doing talks for Bastyr University. He does work for University of Washington, University of Cal Davis, Georgia, so a lot of different stuff.
Yeah, the kicker is you gotta find somebody to pay for this, because it's not all recreational.
Right. So that's what Ryan does. That's what he's starting to do. So, yeah.
My little thought, our present method of testing what we're looking for is so unsophisticated compared to this. It just blows me away when you realize how sophisticated it is. Yet, what you're doing is so unattainable to us because we don't have that kind of money.
So it brings us back to what can you do for $20 a pop?
Exactly. So what we can do is so unsophisticated, it doesn't really give us the answers we want. How do we get the answers we want? We need sugar daddy or something to do this.
So sequencing the water sample, it's $220. It's not outrageous. The database is what's going to be the
Yeah.
expensive cost, and that's going to be funded by a government agency. There's no other way.
So the 220 doesn't deal with the library at all?
It doesn't do any water, but that will get you, that bar draft, those samples, each one. Yes.
Let's get to this issue about comparability in different regions, because there is a thought that there is a pilot that needs to be done. And are you assuming that a useful pilot would have uniform results for lagoons in the Yakima as opposed to Whatcom County? Because there would be a contest to do it right one place rather than to spread the money so far that you don't get a particular answer in any one place.
Right, so that's why that commission budget, they know 150,000 was designated for the west side, really Whatcom County. 100,000 was the east side. To me is, if you're going to do this, and this is the big if, this is unique. Nobody else is applying it this way. We are the only ones, is because of the person, Brian came in, he's done this for his master's, he did this in Peru. And I sat down and we talked, and I said, here's an issue. My background brings me all this information and say, can we apply it here? And we started doing it. We invested over $200,000 of our own money into seeing if this works before we presented anything. So, and we started doing this and started going, we've got an application that we think this could work. So it's taking it, what you're saying is Yakima. So yes, let's do Whatcom County, see how it applies, but you got this $100,000 they want to do Eastern Washington, run it, compare. Is it different? Is it not?
Or I guess it comes down to how much do you think it's going to cost to do the full pilot in Whatcom County? And if it's more than 150,000, perhaps we need to scheme on how to fill that out. And it might be to talk to, was it Hungerford over there that has a claim on that part of the funding?
Yeah.
And suggest that it's better invested in one location to test the.
Right. That's all the stuff in Olympia. They're the ones that decided that. But we've done some preliminary, as we're gaining more information and more feed. Our first initial look was a year-long study, but people are adding to this reference database. So now instead of 8 or 10 locations, I think we got up to 15. So it's changing it. But we felt we could get a lot done for 150,000.
And how long do you think? That's 1 year to collect samples. We'll get results after that year, or
We'll start sequencing month 1. So we'll take we'll take all these samples. We will start extracting DNA, start the sequencing run, and data will come out. We'll be generating data as this goes. So if somebody says, hey, I'm going to start taking water samples at the same time, that's not a problem because we'll always have that data and compare it to these eventually. And like I said, we feel the first 3 months we should be running fecal coliform from these sources with the MF and comparing that using sequencing to see if we do have an indicator with that test. It's been run for years. I've run it since the beginning. So, and membrane filtration, as much as they want to say it's membrane filtration lab to lab doesn't mean it's all run the same way. There's different filters you buy, and there's different medias you buy. I've done the research, I've done studies. The temperature's all supposed to be 44.5. There's a plus or minus with the thermometer. There's a plus or minus with the incubator. The vacuum they use to run the membrane filtration. All those things are different, and there's all a lot of variability. So that's why you see inter-lab testing. Harry Brake, who retired, worked for the Department of Health. Talked to him, gosh, this was probably 15, 20 years ago when we were going over fecal coliform tests. And he goes, if I got a sample that's 10 and a sample of 1,000, it's the same number to me. He said it's log 2, log 10 to the 2. So he said it's the same number to him. So, yeah, 220 versus 210, that's the same number.
Can I just want to follow up with what you said, Dory, about the, just fecal coliform grab sampling being unsophisticated, which there is a lot of value in this, and, but there's also a lot of challenge too, like the fact, I mean, it takes months to process these samples, and I guess I would just like to keep people optimistic, the fact that, yes, it's kind of like the mallet of a toolbox, and we've been using it for a long time, and maybe it doesn't give us all the questions, all the answers that we want, but when we look at like improving trends over all the watersheds in the last, many years, it's helping, and it's allowing people to like bracket areas and actually find sources, and I just think we should keep that in mind as we it's easy to look at this and say like, wow, this is a gold standard. What we're doing is, scuffing our feet around, but we've made a lot of good progress.
Right, I'll say this, is there's different tools. Each tool has its place. Fecal coliform monitoring, using what you use now, has its place. There's different techniques that I think I would look at versus the membrane filtration, but that daily, weekly, monthly monitoring has a place. It is needed because it's quick and easy. This is just another tool that says, okay, we've got an issue, let's go really find out what it is. And it's just another avenue to help define things that's happening in the watershed. But is it a daily run test? I don't think it'll ever be a daily run test. It's not going to be a weekly run test. You may find problematic areas and say, oh, hey, we're going to monitor this weekly, and we will get this technique down to where it's not taking months, it wouldn't be a week. I'm running samples for other clients using sequencing that has nothing to do with water. We're IDing herbs, fish, bacteria. We're doing all kinds of different things. We turn those around in 4 or 5 days. So it's not a long process, but the application, you have to understand, is I don't expect this test to replace fecal coliform. It's a test to help fecal coliform testing.
With that, I'd like to close the meeting, but if you'd like to stick around and talk with people one-on-one.
Sure.
At the hour?
I know like the Hearst, we had things that we talked about doing, other projects we look at. I used to run what's called MPA testing for wells. Back in the 90s, early 2000, I was one lab in, probably there's 10 of us in the US. I got trained by Manchester. So what we did is, what the state mandated is any well within 200 feet of a body of surface water had to test by microscopic particulate analysis to see if they were under the direct influence of that surface water. Did hundreds of these samples. This tool is way better. Because what we did is we looked for algae indicators, we looked for insect indicators, Cryptosporidium giardia. This was taking a 100-gallon filter, concentrating it down to a pellet, and looking at that pellet, and it could take 50 slides to get the right quantitation. I could have done this and been done.
Actually, if you can show continuity between groundwater and surface water, you may have a booming business.
Right.
Yes.
Right, but that is possible with this method. I did MPA. EPA developed the method. Jay Vasconcelos with Manchester Lab, he's retired now. Stephanie Bailey, and they all work with Stephanie. There's the two Stephanies at EPA Manchester. The only one, Stephanie Bailey's the only one left, or Stephanie Crane.
Yeah, that's right.
What?
Stephanie Bailey's left.
Stephanie Bailey, and Stephanie, she was the best.
Stephanie Harris.
Harris, yes. Stephanie Harris was the one who retired. So I worked with all of them with MPAs, ran tons of them. So that is a possibility, is with this Hearst decision, there is an application.
Can we all give?
Did you get through all your stuff?
Yeah, the next one, just